Welcome friends to a new session on genetics. In this session, we will discuss extrachromosomal inheritance, mitochondrial genetic system, organellar genetics, cytoplasmic or extrachromosomal inheritance. Part 1. Let us first see about mitochondrial genetics. In 1970, Lynn Margulis published the book Origin of Eukaryotic Cells that effectively revived the long-standing idea that mitochondria and chloroplast evolved from free-living bacteria via symbiosis within a eukaryotic host cell. Various endosymbiotic models proposed over the years have been comprehensively evaluated, while the debates surrounding the endosymbiont hypothesis that traces the development of ideas regarding organelle origins. Yet, the evolution of mitochondrial genome has taken radically different pathways in diverse eukaryotic ancestry, and the organelle itself viewed as a genetic and functional mosaic, with the bulk of the mitochondrial proteome having an evolutionary origin outside Alphaproteobacteria. New data continue to reshape our views regarding the evolution of mitochondria, particularly raising the question of whether the mitochondrion originated after the eukaryotic cell arose. The mitochondrion is a double membrane bound organelle found in most eukaryotic cells. The word mitochondrion comes from the Greek mitos means thread and chondrion means granule or grain like. Mitochondria range from 0.5 to 1.0 micrometer in diameter. There is a considerable variation seen in the structure and size of mitochondria. They are not visible unless specifically stained. Mitochondria are known as the powerhouse of the cell because they generate adenosine triphosphate ATP, used as a source of chemical energy. Although most of the cell's DNA is contained in the cell nucleus, the mitochondrion has its own independent genome. Further, its DNA shows substantial similarity to bacterial genomes. We know that, like the chloroplast, the mitochondria of both animals and plants also contain DNA. But unlike chloroplast genome, mitochondrial genome, that is mtDNA, shows fairly large range of variation in size and in its proportion in the cellular DNA. It varies in size from 15 to 19 kilobase in Diptera, mammals, amphibians and fishes, from 15 to 176 kilobase in protists and fungi, from 200 to 2400 kilobase in higher plants. However, the number of mitochondria per cell is large, although relative to the nuclear DNA, usually the mitochondrial DNA still represents only about less than 1% of cellular DNA, except in yeast. In yeast, mitochondrial DNA may represent as high as 18% of the cellular DNA, each mitochondrial genome being 85 kilobase. Some details of mitochondrial DNA are shown in this table. Like chloroplast DNA, mitochondrial DNA is usually circular, but in ciliate protozoans, Chlamydomonas reinhardti, and also in some yeasts, the mitochondrial DNA is a linear molecule. The genetic role of this DNA is also understood now. The most important experiments on mitochondria were done by Boris Efrusy in yeast, which was initiated by the discovery of Petite mutants. Subsequently, mitochondrial DNA was studied in several organisms, including plants and human beings. Let us examine about Petite in yeast, a mitochondrial character. The Petite mutants in yeast fail to grow on carbon source like glucose and produce smaller colonies when grown on sugars like glucose. This is a difference which can be observed only in the presence of oxygen, so that these peptides actually have a defective aerobic respiratory mechanism. These peptides differ from wild type called grande and are characterized by 1. Their insensitivity to inhibitors of aerobic pathways, 2. a number of changes in the respiratory enzymes in mitochondria and absence of cytochromes A, A3, B, 3. lack of sustainability of Petite mitochondria, and 4. incompletely developed state of mitochondria. The Petite mutants can be segregational following Mendelian segregation and therefore presumably controlled by chromosomal genes, or may be more often vegetative, that is non-segregational, or extrachromosomal. The genetic basis of Petite character is a cytoplasmic factor Rho, which may be absent or defective in peptides. The vegetative peptides thus can be neutral, which completely lack Rho factor, or they can be suppressive having a defective Rho factor. The neutral peptides are not transmitted, while suppressive peptides are transmitted to a fraction of vegetative diploid progeny. Suppressiveness varies from 1 to 99% peptides in various strains. Two lines of evidence suggest association of Rho factor with mitochondrial DNA. 1. Ethidium bromide, which induces peptide mutations with 100% efficiency, causes degradation of mitochondrial DNA after neutral peptides have actually been found to lack mitochondrial DNA. 2. Suppressive peptides contain mitochondrial DNA, which is often grossly altered in base composition with respect to wild mitochondrial DNA. Let us see about Poky Neurospora, which is a mitochondrial character. A Poky mutant in Neurospora differs from wild type in the following attributes. 1. It is slow growing. 2. It shows maternal inheritance. And 3. It has abnormal cytochromes. Of the 3 cytochromes, cytochrome A, B and C found in wild type, cytochrome A and B are absent and cytochrome C is in excess in Poky mutant. In reciprocal crosses, Poky character shows maternal inheritance. Poky female crossed with wild type male, all progenies are Poky. Wild type female crossed with Poky male, all are wild type. Although other marker nuclear genes show 1:1 segregation, following evidences suggested that Poky may be located in mitochondrial genome. 1. Slow growth may be due to lack of ATP energy, and source of this energy is mitochondria. 2. Cytochromes in Poky differ from those in wild type in quantity and quality, and these cytochromes are found in mitochondria. Now we are going to discuss about mapping of mitochondrial genes in yeast. The mitochondrial genes in yeast is depicted in this table, and it have been mapped using a variety of techniques, some of them similar to those used for chloroplast genes. These techniques have been extended for mapping mitochondrial genes in other organisms also, and will be briefly described. A. Recombination mapping. Since there are no chloroplast in yeast, all genes showing uniparental inheritance can be attributed to mitochondria. Uniparental inheritance may be established when two mating types are fused, the fusion product being a diploid, that is 2N, and also a cytohet, that is cytoplasmically heterozygous. If diploid cells are allowed to divide mitotically, that is by budding, segregation and recombination occur, but when this budding is followed by meiosis. Meiosis can be induced by shifting the cells into a special medium, and because of this all ascospores in an ascus are one type with respect to mitochondrial genes, although they show segregation in the pattern of 1:1 for nuclear genes, including the gene for mating type. This is illustrated in this figure using a cross between erythromycin resistant and erythromycin susceptible. Using the above principle, if a dihybrid cross such as erythromycin resistant and spiromycin resistant is made with erythromycin sensitive and spiromycin sensitive, and diploid cells are allowed budding followed by sporulation, all asci showed lack of segregation, and this is shown in this figure. And the following results could be obtained: erythromycin resistant and spiromycin resistant 63 tetrads, erythromycin sensitive, spiromycin sensitive 48 tetrads, erythromycin sensitive, spiromycin resistant 7 tetrads, erythromycin resistant, spiromycin sensitive 1 tetrad. Among these 4, 2 classes are recombinants, but they result from several rounds of recombination during budding as in phages. Recombination is also influenced by a genetic factor omega not found in all yeast strains. Nevertheless, recombination data like the above have been utilized for mapping. Recombination generally never exceeding 25% as against 50% for nuclear genes. B. Mapping by Petite analysis. In the above technique, Petite character is artificially induced in wild type. It is also called as grante, carrying several mitochondrial marker genes showing resistance to 4 different antibiotics: erythromycin resistant, chloramphenicol resistant, oligomycin resistant, spiromycin resistant. The peptides so induced are screened for retention or loss of these mitochondrial genes. The peptides cannot be tested directly for drug resistance and therefore are first rescued by fusion with drug susceptible grante cells, so that if on budding some of the fused 2N cells are drug resistant, this would mean that peptide was resistant, and this is clearly shown in this figure. If frequencies of genes which are retained or lost together are compared, this would give an idea about the strength of linkage. C. Overlapping of mitochondrial DNA segments in peptides. In the above analysis, peptides can be identified which retain different sets of mitochondrial genes. For instance, suppose peptide A retains chloramphenicol resistance, erythromycin resistance, parmomycin resistance, and peptide B retains chloramphenicol resistance, erythromycin resistance, oligomycin resistance, antimycin resistance. Now, DNA can be extracted separately from A and B and hybridized with the DNA from wild type cells. Suppose amount of DNA hybridized is A units with the peptide A, B units with the peptide B, and T units with A plus B together. Now, if T is less than A plus B, then there must be an overlap. H is equal to A plus B minus T. In this overlap region, H must be found genes retained both in peptide A and peptide B. The overlap H should also represent the maximum distance between chloramphenicol resistance and erythromycin resistance. This will allow placement of markers into map segments of defined size. and you can see this phenomena in figure. A library of peptides is thus now available with various genes retained and different peptides related due to specific overlap regions. An interesting feature of peptides is that the amount of mitochondrial DNA in peptide and Grant A is almost equal. This is due to amplification of a small DNA segment which is retained in peptide. This you can clearly see in this figure. This equality of DNA does not interfere with the technique of overlapping mitochondrial DNA segment utilized for genetic mapping. D. Mapping by restriction enzyme analysis. Restriction fragment analysis has also been utilized for mapping, as has been done for chloroplast also. In this particular figure, the restriction map of yeast mitochondrial DNA is shown, and the figures in the outermost circle represent kilo base pairs of DNA. Inner 4 circle shows restriction site for 4 enzyme combinations. The tRNAs, rRNAs and other RNAs can be hybridized with the restriction fragments to identify mitochondrial DNA fragments carrying the genes. The limits of these fragments can be narrowed using fragments produced by different restriction enzymes separately for hybridization with the same RNA. Restriction mapping may also be combined with techniques of peptide analysis and overlapping DNA segments as described previously. This allows us to locate physically the genes for drug resistance on specific restriction fragments. The circular genetic map of yeast prepared following the techniques already described is presented in the figure to the left, and to the right the genetic map of human mitochondria is shown. In the maps of circular yeast and human mitochondrial DNAs, both the gene products and gene symbols are shown. Black boxes show regions of mRNA or rRNA synthesis. Small circles are regions of tRNA synthesis. Now, let us examine human mitochondrial DNA. In human beings, mitochondrial DNA is smaller in size, just 1.66 kilobases, and the complete nucleotide sequence of this DNA is now known. Human mitochondrial DNA encodes 13 protein coding genes that are subunits of the oxidative phosphorylation system and 22 transfer RNAs and 2 ribosomal RNAs. This is a remarkable example of complete genetic map obtained without any genetic analysis, and you can see the following in this figure. Now, when we examine about plant mitochondrial DNA, it has evolved rapidly in structure but slowly in sequence. The mitochondrial DNA of higher plants is larger and more complex, with 200 to 2500 kilobases, and is present in many different molecules. The structure of mitochondrial DNA in several flowering plants is under investigation now. It seems that in plants, mitochondrial DNA molecules of variable size are available, making the study relatively difficult. The size of the mitochondrial genome is quite variable. Example, in Oinothera it is 195 kilobase. Coming to the case of Sia maize, it is 570 kilobase. The circular mitochondrial DNA of Marchantia polymorpha contains 1,21,024 base pairs, which make up some 128 genes, which includes duplicate genes encoding 4 subunits of the rRNA, that is 23S, 16S, 4.5S and 5S subunits used by the chloroplast. 37 genes encodes all the tRNA molecules. 4 genes encodes some of the subunits of the RNA polymerase. A gene encodes large subunit of the enzyme Rubisco. 9 genes for components of photosystem 1 and 2. 6 genes encoding parts of the chloroplast ATP synthase. Genes for 19 of the 60 proteins used to construct the chloroplast ribosome. Now, let us make a comparison of mitochondrial DNA in yeast and mammals. We have seen that the mitochondrial DNA of only a few individuals has been analyzed in detail so far. In the case of yeast and mammals, however, some clear picture has emerged. The sets of known genes in these two classes of organisms are identical except some differences, as shown in this table. For instance, yeast has 25 tRNA species, while mammals have only 22 tRNA species. Further, the genes for subunit 9 of the ATPase complex and those for a ribosome associated protein, though present in yeast, are absent in mammals. Mammalian mitochondrial DNA is transcribed into a single transcript from the major coding strand, and individual products are generated by RNA processing. In yeast, rearrangements can occur in mitochondrial DNA rather frequently. A substantial fraction of the genetic material in mitochondrial DNA in yeast as well as in mammals is represented by unidentified reading frames. In short, we call it as URFs. Most of the yeast URFs are contained in introns of split genes and appear to code for protein required for mitochondrial RNA splicing, the so called maturases. The URFs of mammalian mitochondrial DNA code for proteins of unknown function. Now, let us see about unidentified reading frames, URFs and protein coding genes in mitochondrial DNA. In animals and particularly in mammals, 13 reading frames coding for proteins ranging from 70 to 610 amino acids have been identified. These are in addition to the genes for ribosomal RNAs and tRNAs. 5 of these 13 reading frames code for cytochrome C oxidase subunit 1, 2 and 3, ATPase subunit 6 and cytochrome B. The remaining 8 reading frames, representing 60% of the protein coding sequences, pertain to the so called URFs whose products have not been identified with any protein of known function. Nothing is known about the physiological role of the products of mammalian mitochondrial DNA URFs, except that they are hydrophobic proteins associated with the inner mitochondrial membrane. URF A6L is perhaps homologous to AAPL coding for a protein of 48 amino acids, which may be a part of ATPase complex. Let us also see about unidirectional replication of mitochondrial DNA, that is D loops. Another interesting feature of mitochondrial DNA is its unidirectional and highly asymmetric replication. The daughter L strand starts synthesis when 2/3 of H strand is already synthesized. The position of origin of synthesis of H strand is marked with the displacement loop, that is D loop. Now, coming to the fact of evolutionary origin of eukaryotic mitochondria from prokaryotes. A comparison between the properties of mitochondria and bacteria has shown similarities which indicate evolutionary relationship. Some of these similarities are listed in the following table. On the basis of these similarities, it has been suggested that mitochondria might have evolved due to symbiotic association of bacteria with cells of higher organisms. In the table, the last three similarities between mitochondria and bacteria are rather very conspicuous. These include size and shape of DNA, there being circular DNA in both. The DNA in eukaryotic chromosomes is very different than circular DNA found in mitochondria in the same cells. Further, the protein synthesis in eukaryotic cytoplasm is inhibited by cyclohexamide and not by chloramphenicol as in bacteria and mitochondria. This property has been utilized to stop preferentially the protein synthesis in cytoplasm by cyclohexamide and thus marks the protein synthesized in mitochondria or chloroplast, both responding in the same manner to cyclohexamide and chloramphenicol. Similarly, protein synthesis in mitochondria and chloroplast can be preferentially stopped by chloramphenicol and the protein synthesized in cytoplasm can then be identified. The property of protein synthesis mechanism is similar in mitochondria and bacteria due to similar ribosomes, that is 70S. Now, let us summarize the following discussions on mitochondrial genetics. Both animals and plants also contain DNA. Mitochondria and chloroplast evolved from free living bacteria via symbiosis within a eukaryotic host cell. Unlike chloroplast genome, mitochondrial genome shows fairly large range of variation in size and its proportion in the cellular DNA. The number of mitochondria per cell is large, although relative to the nuclear DNA. Usually, the mitochondrial DNA still represents only about less than 1% of cellular DNA. Mitochondrial DNA was studied in several organisms including plants and human beings. In yeast, Petite is a mitochondrial character, that is, the Petite mutants in yeast fail to grow on carbon source like glucose and produce smaller colonies when grown on sugars like glucose due to the presence of oxygen. Another mitochondrial character is Poky Neurospora. A Poky mutant in Neurospora is slow growing, shows maternal inheritance and has abnormal cytochromes. The mitochondrial genes can be mapped using a variety of techniques such as recombination mapping, mapping by peptide analysis, overlapping of mitochondrial DNA segments in peptides and mapping by restriction enzyme analysis. Now, I will give you certain assignments based on the discussions we had till now. First one, discuss the evidences suggesting the role of mitochondria in cytoplasmic inheritance. 2. Discuss the techniques of mitochondrial mapping in plants and animals. 3. Briefly explain the physical basis of hereditary factors located in the cytoplasm. 4. Write short notes on mitochondrial DNA. 5. Discuss briefly the similarities between mitochondria and bacteria suggested the origin of mitochondria from bacteria. Let me give you some references for your further reading. 1. A book entitled Cell and Molecular Biology, authored by D. Robertis and Robertis, published by Leah and Febiger, Washington, in the year 1991. 2. A book entitled Genetics and the Origin of Species, authored by Dobensky, published by Columbia University Press, New York, in the year 1961. A third book is entitled Principles of Genetics, authored by Gardner and Nestad, published by John Wiley, New York, in the year 1984. Fourth is book entitled Genes, authored by Levin, published by Oxford University Press, London, in 1994. A fifth book is entitled Polyploidy, authored by Lewis, published by Plenum Press, New York, in 1980. A sixth book is entitled Chromosome Technique, Theory and Practice, authored by Sharma and Sharma, published by Aditya Books, New Delhi in 1980. A seventh reference is entitled Principles of Genetics, authored by Sinnott, Dunn and Dobzhansky, and published by McGraw-Hill, New York in the year 1958. I will also give you certain websites. One is www.genetics.org. A second one is www.theagricos.com. Now we have come to the end of this session. See you in another session with a new topic. Until then, bye.